Hey, boys and girls. So we're here for the very last part of the hot springs hypothesis paper, part 5, I believe it is. And Scott found a video that came out today, right?
The video press, yeah.
And 57,000 views or just came out.
And welcome.
Whoa.
Oh, wait a minute. I had something in my tab.
In fact, I had one earlier this morning.
Yeah, don't
You're actually playing the video, Scott. Let me know when you unmute yourself when you're done playing the video.
Okay, sorry about that.
Okay, that happens. Okay, so we thought we'd play a little bit of this video because it's on the hot spring. Does she refer to this paper?
I don't think she refers to the paper, and she doesn't really refer to Damer or Deamer, but she does refer to it as the hot springs hypothesis. And so it just shows where it's, of course, their thoughts were published in Scientific American a few years ago. So I think this is just something that's gradually becoming more and more popular because of the water paradox problem. The fact you have all that salt water, there's so much water in the ocean, and then ultraviolet rays tend to break up other than great concentrations of the organic material they need. So
yeah.
But anyway, it's something to listen to.
Okay,
we'll give a review.
We'll listen ahead.
Enough concentrations and to be exposed to sunlight for that crucial UV component. Some scientists believe that this life-giving reaction had to have occurred in environments like small pools. And next, building on the work that produced the nucleotides, in 2015, Sutherland and his team were also able to produce the building blocks of proteins and fats. And that's a big step because now with nucleotides and protein and fat precursors, we've got all the moving parts needed to get life going. And in 2020, other teams have found even further answers to the water paradox. Researchers created conditions where almost proteins could form and survive during wet-dry cycling, which is exactly what it sounds like. The molecules are allowed to dry out for some time before they're exposed to water again. And the researchers discovered this process is selective. Only the molecules we would expect to see on an early Earth withstood this cycling and were actually shaped by it. This wet-dry cycling has even shown to essentially select for more complex combinations of nucleic acids inside little protective lipid bubbles that help those nucleic acids make it through dry periods. And scientists think this may be a precursor to something like a cell. And all of this lends even more support to the idea that the body of water from which life may have sprung would have to be shallow and prone to drying out from time to time. Imagine all of life being descended from some stuff growing in a puddle. It's also worth mentioning here that not everyone is in agreement on this. Although there are commonly agreed on necessary original ingredients, some teams are still looking into deep sea vents as a possibility.
Did she mention Sutherland?
Yeah, right as you started it, she talked about Sutherland and that experiment that ended up with nucleotides. Yeah. But she mentioned him before that as well.
Yeah, he has a 2017 paper on opinion studies on the origin of life that's referenced in Hot Spring Hypothesis paper. So anyway, but she may link this paper. I'm not sure. I would have to look at the link. She doesn't name the paper.
Yeah.
But anyway.
For the birthplace of life, while others think hot springs are a likely contender. And why is any of this important anyway? Aside from answering existential questions about where the heck we come from, a better idea of how life originated here on Earth gives us clues about what it might look like or where to look for it on other planets too. For example, in February of 2021, the Perseverance rover successfully landed in Mars's Jezero crater, which seems to have once been an ancient lake. And given all of this research, the crater is now a prime suspect for signs of life on the red planet. Now, it'll be years before the samples return to Earth, but maybe
Okay, so I don't want to play anymore.
Right, yeah, that's fine.
I did link this in the description, so you can go ahead and watch it. So everyone can watch it.
Very cool.
Yep.
But that shows kind of that kind of thinking is making it out there, and it just seems like this video has to have been informed by Damer and Deamer's paper.
Yeah. Yep. In a second here, I hit the wrong button. Gotta get the right button to look at the paper. So I think it's Damer and Deamer.
Damer
Yeah.
and Deamer.
Deamer. Okay, so back to where I'm supposed to be. Where am I supposed to be?
I don't know, but right now you're in the reference section.
Okay, I think I'm here. That's where we left off.
There we go. Network effects.
Here we go.
Let's see. Network effects and other evolutionary properties of the progenote. That idea of the progenote is an important one, and it crops up basically for the rest of the paper.
Yep.
The demonstration of the existence of a complex non-enzymatic metabolic cycle, such as the reverse citric acid, would be a major step in research on the origin of life, while demonstration of an evolving family of such cycles would transform the subject. We agree with, I'm going to say Orgel's statements that the emergence of, oops, sorry about that. I'm getting a notification that you've gone live.
Oh, that's important to know.
Yeah. Let's see. Statement of the emergence of closed cycle metabolic reaction networks remains a key unsolved problem for the origin of life. Life depends on highly evolved networks which harvest energy to synthesize products and to coordinate and regulate reactions. In parallel with the initial bootstrap of stable protocells and stepwise selection of functional pollinators within the kinetic trap is a necessary emergence of a network effect. Computer networks, such as the internet, are made up of services based on protocols that enable packet delivery, error correction, dynamic table lookup, and pathway optimization.
Uh-oh, analogy.
I know, it's an analogy alert. Analogy. Not to be taken literally.
Okay, each.
You want to read it? Go ahead.
Oh, no, I moved it on you, but
Okay, yeah, we're at each. Each service is simple on its own and often described by a few programmatic steps. Yet the emergent network, composed entirely of closed cycles, attains a high degree of operational robustness and adaptive capability. This capability was specifically designed into the original ARPANET, which grew into today's highly robust and scalable internet, thanks to Al Gore. No, that's not part of it. I just added that. The network effect codifies the old adage, the whole is greater than the sum of its parts. During the evaporation phase of wet-dry cycles, protocells aggregate into a hydrogel within which a network effect can be initiated. And, I think that idea of a hydrogel is something that the hydrothermal people, the ones who, theorize and scratch their heads over deep sea hydrothermal vents also postulate. So it's kind of a foundational idea to both hypotheses. Such gels retain moisture and concentrate nutrients, providing for the selection and sharing of the additional polymer functions, such as pores required for solute transport, supporting emergent metabolic cycles within protocells. Recent work demonstrated radiolysis reaction networks connecting geochemical reservoirs to the production of carboxylic acid, amino acids, and ribonucleotide precursors. These networks exhibited hierarchical organization and families of closed-loop commons to biological systems. Such networks of interacting radicals driven by UV and other radiation sources operating within protocell aggregates could have provided the initial cycles which Orgel sought. Something with the
What?
Uh,
Did you
can
miss?
Yeah, the first part of that sentence, I didn't catch it.
Well, just you want to cell size compartments.
Yeah, something cell size compartments whose interiors enable molecular crowding, and when aggregated together, they provide an ideal environment for selection through passive forms of protocell competition. Furthermore, an aggregate would tend to retain the components of disrupted protocells, thereby selecting for surviving protocells that can absorb and conserve the components in a primitive form of heterotrophy. Heterotrophy?
Heterotrophy. Okay. Yeah, that's good.
So this is an important part because you have these reactions going on. They get individually locked within these bubbles of lipids, and if they get destroyed, then what items have developed so far chemically just will get lost, torn apart in the water solution. But when the water is evaporated, those same molecules just kind of get overlaid on the top and the bottom and the side with those dried out lipids. So they're protected and they're tightly close together with other molecules and particles to where they retain the ability to continue with whatever development they're going to take on. That's what I took away from that. And that seems to be a very important idea of preserving those compounds from the effects of the water paradox.
Yep. And I've did a little thinking today and searching for when the protocell expands so that it's in its spherical form. Then there is the problem of getting transmembrane proteins and basically compounds or channels, that sort of thing.
Mm-hmm.
Now, there's some of that stuff happens naturally for absorbing ions and things like that, but more complicated things, I'm wondering exactly because when proteins are created, they're sort of inserted into the lipid bilayer as they're being created. And that's how we end up with these. There's a really neat mechanism for ending up with a alpha helix protein that crosses the membrane 7 times. And that creates a particular kind of receptor, which we have about 900 of them in our body. But anyway, I'm looking for different ways that those things can be integrated. But I think at this stage, we're in the meltdown build-up phase, this cycle.
Yes, right. Yeah,
we're not quite there yet. And when it breaks down into these lamella and gels, most certainly at that point, these proteins could be inserted across the membranes. So if there are some to insert. And it takes about 17 to 22 amino acids to cross a bilayer membrane. So, and they only have to be hydrophobic. They don't have to be any particular kind of amino acid. So glycine and anyway. Okay, so where are we? We
need to go up a little bit more.
I'm sorry.
Okay, no, there we go. What follows is a conjecture. So let's underline that so that if anybody is reading it with a mind towards finding fault with it, we're all aware that what follows is conjecture or speculation, kind of supposing things happening based on physical realities that have been proven in the lab.
You mean they're not claiming it's a fact?
Not necessarily. Not necessarily.
Like Dr. Knack says? Not
like Dr. Knack.
Okay.
However, we believe it has value because a conjecture becomes a working hypothesis if it can be falsified by experiments, several of which are proposed in Section 6. A protocell aggregate can be considered to be a prototypical form of niche construction as described by Odling-Smee et al. Deamer extended this concept to prebiotic conditions in which protocell populations form moist gel aggregates during the drying process that protect individual protocells and their polymer cargoes against shear forces, pH extremes, and temperature changes. A gel could also maintain a stable concentration of ionic solutes, thereby protecting protocells against osmotic stress.
Okay. You want me to take over?
Sure.
I don't think I'm ready.
Well, I
Figure
can do
eight.
it.
Figure eight.
Okay,
Figure eight left. Where's figure eight? Oh my goodness, where's figure eight? Left illustrates how the close aggregation of compartments allows metabolic products to diffuse within protocell populations. Figure eight left, it's not, there is no.
I think it's on the bottom.
Yeah, so where am I?
If you scroll down.
Am I on? There it is.
Oh, there you
figure
go.
Eight. Yeah. So if we look at figure eight, what do they say it's left? Just an interesting
Probably
little figure.
Originally it was, or maybe that's just a mistake they forgot to edit.
Yeah. Probably originally was. Okay, figure eight. Like, such products and whole complexes of functional polymers can be mixed throughout the aggregate and thereby benefit the entire population, enabling a mechanism to support inclusive fitness. I want to highlight that. This property also models the network effect described earlier and is a primitive version of horizontal gene sharing characteristic of all microbial communities today. And as introduced in the previous section, an aggregate of protocells is a candidate progenote originally described progenote.
Yeah, progenote.
Described by
little lambs eat ivy.
Wosenfox and Woese, 1998, in which its member protocells are engaged in the collective process of evolving the relationship between phenotype and genotype. We introduce the name progenian to describe this transitional evolutionary period. Interesting. There is no rock record left by protocells or their progenote aggregates, so the term cannot specify a geological era, but instead refers to the stages of chemical evolution from simple self-assembling protocell populations into living microbial communities. For example, early in the progenian
Progenian, yeah, I think that's right.
Consortia of protocells would emerge, which are capable of growth and adaptation through phenotypic properties, emerging by simple selection from random sequences. A middle division of the progenian would begin.
Dun, dun, dun.
By the way, Immutable might be joining us. I'm not sure. I haven't heard from him.
Oh, good.
He doesn't. He does have a link whenever he's ready. Okay, with the first protogenetic system, perhaps initiated by simple templated replication. The late stages of progenian would be characterized by sets of increasingly functional polymers that are duplicated by a primitive replication and translation system and collectively passed on to subsequent generations of protocell populations. So Demeter's hypothesis would fit right in to these middle and later stages. So, and maybe even the beginning a little bit. Throughout this period, passive self-assembly operating on the scaffold of wet-dry cycling is gradually replaced by systems of more efficient active polymers interacting in networks, giving protocells an increasingly coherent biological identity. Now, the other thing that might be happening during this period is a transition from those hot volcanic pools to cooler pools that are, sort of downslope. And
right.
So there'd be a continuum of different environments going all the way from basically the hot pools all the way down to cool pools down to the ocean, in fact.
Yeah, and on any given geologic area, you could have several of these streams that gather in pools that trickle on down to a cooler environment and eventually to the seaside. So you take the 3 or 4 that could possibly exist on one island, multiply times the number of islands, and that increases the number little labs, little experiments that are taking place all at once until that right combination of, you know, of protocell with the right kinds of nuclei in it. I say nuclei, I just mean organic configurations inside the protocell that would, you know, help preserve itself, help preserve the integrity of that little cell, and then eventually be able to reproduce itself.
Yep. So where did I leave off? Did I leave off with identity? Or did I can't
think
remember.
throughout the cell, passive cell assembly.
Oh, okay. Throughout this period, passive self-assembly operating on the scaffold of wet-dry cycling is gradually replaced by systems of more efficient active polymers interacting in networks, giving protocells an increasingly coherent biological identity. A key question is how the progenote served as an evolutionary environment to transform simple, passively budding protocells into actively dividing living cells. It is reasonable to assume that the first functional polymers operated most effectively while bound to a protocell's membranous enclosure rather than dispersed within the dilute interior. Now that's interesting. So bound to that membrane. This binding has been shown to stabilize the membrane against stresses. I think last time I found some papers that talk about how proteins will stabilize those lipid bilayers, even though they could be of just one type of substance, basically. They're more stable, I think, if you have multiple types of lipids or phospholipids, but they still work. This binding has been shown to stabilize the membrane against stress, and would also enable sets of polymers to become more crowded on the two-dimensional surface and interact more, and interact more easily. Recently, Cornell proposed that a co-localization sets up a positive feedback loop in which amino acids bind to self-assembled fatty acid membranes, resulting in membrane stabilization and leading to more binding. And that high local concentrations of molecular building blocks at the surface of fatty acid membranes may have aided the eventual formation of proteins, just as the viable operation of protocells would have been dependent on binding and interactions between enclosures and polymers. The process of their continued evolution would have been dependent upon the protective and nutritive environment of the surrounding progenote aggregate. The long evolutionary climb to cell division would have been a stochastic process driven by innumerable tests and setbacks. Aha. Failure of a key process within a protocell dwelling separately in solution might result in disruption and dispersal of the entire cargo of polymers, whereas such faults would not be so deleterious if they occurred inside a communal matrix. So they would, there would be just horizontal transfer. Mm-hmm. Challenges arising from unreliable metabolic circuits within a protocell can be mitigated through networks of resupply of materials and energy from other protocells operating in the surrounding aggregate. Hang on a second here. Hairball. At some point, evolutionary innovation. You gotta have cats, man. If you want to have fun, you gotta have cats. At some point, evolutionary innovations converge to where protocells are selected, which begin to produce subdivisions of their own compartments, possibly beginning as a simple adaptation to bud and eject vacuoles containing waste. This is all fascinating. In the case of I'm going down to another page. Mm-hmm. Case of a failed compartment division resulting in disruption of the entire protocell, valuable functional polymers would still be retained and reused by other protocells in the surrounding aggregate. The final transition to life could have been facilitated through the chance combination of a fortuitous division of a compartment occurring just after the duplication of two sets of membrane-bound polymers, each pushed into opposite ends of an elongated protocell. In this first example of binary fission, the time triggering of a transverse division of such a compartment would securely deliver duplicate sets of minimally viable polymers into daughter cells. Through the resulting innovation of cellular reproduction comes a vertical inheritance of traits and the transition out of the progenian into the era of living microbial communities. As is obvious from the above example, a key evolutionary property of progenomes is that they can support attempts at the failures of risky multi-step processes, such as reproduction without the loss of genetic material. This communal support system thereby permits the incremental writing of the complex code of life. And at this point, we do not have DNA yet, as far as we know, but we could.
Correct.
I mean, it could have popped up. Okay, I'm going to let you take over for a minute.
The speciation and specialization of increasingly robust living cells would allow communities to scale steeper gradients of environmental stresses and colonize more niches on the landscape. During the progenian, there would undoubtedly have been innumerable starts and failures, not only of individual protocells, but also of entire progenote communities. However, we posit that a landscape of large numbers of interconnected hydrothermal pools continuously driven by regular cycles of energy and supplied by feedstocks of organic material over tens of millions of years had the combinatorial power to code the operational kernel of life. Eventually, individual cells will become sufficiently advanced to survive and reproduce freely in solution, detached from their original communal complexes. Wider distribution and the competition for resources of communities and free living cells would then extend the tangled branches of early life along the path to the cohort of last universal common ancestors.
Okay. Now we're on section 6, and we're going to try to cover that tonight too. Let's see. Let's look ahead. Where are we at here? We're on which page? Are we on that page? We're on that page. We're on that page. Okay. Oh my. What have I done?
I don't know.
What have I done?
Yeah, here we go. That's where we are.
Okay, I'm trying to figure out what we got left here. So we're on 444. We got 3 pages to go tonight. So
only 3?
Yeah, if we say if we want to finish, we've got.
Yeah, that's fine.
It looks
that's
like
fine. I'm committed to finish this thing tonight so that we have that sense of completion.
Yep, sense of completion is good. Okay.
Okay, a roadmap for testing the hypothesis. Clever phrase ahead. Origin of life cannot be discovered, it has to be reinvented. Albert Eschenmoser. We interpret Eschenmoser's statement that scenarios for the origin of life ought to quickly move beyond paper proposals to become full hypotheses, tested both in the laboratory and in much more complex environments of natural analogs. Deamer et al. 2019. For any hypothesis, a quick empirical falsification is preferable to endless speculation. Yet life is such a complex phenomenon that falsification is not as simple as it can be in physics. Workers in the field must perform multiple experimental tests and use the weight of accumulating evidence to measure the plausibility and explanatory power of any scenario.
I want to mention. Okay,
go ahead.
I want to mention the weight of accumulating evidence, which is why they do purified, controlled laboratory tests. That is the accumulating evidence we're looking for, each step of it, right?
Yeah.
And they mentioned that we should test it not only in the laboratory, but in more complex environments, which is what Deamer and Damer are doing. So anyway,
exactly.
Go ahead. Okay.
Platt's article, Strong Inference, published in Science, 1964, advises experimenters to devise alternative hypotheses and a crucial experiment with the alternative possible outcomes. Carrying out the crucial experiment to achieve a clean result will exclude one or more hypotheses.
Can you draw that out? I'm still working on it. Sorry. There you go.
Following language describes a critical experiment that can be applied to test any origin of life hypothesis. Confirm or falsify a non-enzymatic process by which catalytic and replicating systems of polymers are produced under plausible prebiotic conditions that also support cycles of combinatorial selection in which encapsulated sets of these polymers emerge and begin to evolve. I'm taking a breath here.
Okay.
Proposal,
Go ahead. Go ahead.
proposal for an origin of life in salty ocean water at hydrothermal vents or in fresh water at hydrothermal fields can be considered to be alternative hypotheses. We propose the following 7 tests that can support or falsify the hot spring hypothesis. Number 1, a laboratory observation must also work in the natural conditions it is intended to simulate. In the laboratory, we have demonstrated that in the presence of membrane-forming lipids, wet-dry cycles can drive polymerization of mononucleotides, followed by encapsulation of products in membranous compartments upon rehydration. This experiment was recently carried out in a hot spring setting near Rotorua, New Zealand. The results were positive, but are preliminary, and the experiments need to be repeated. So in other words, they got some good indications there. The wet-dry synthesis of RNA-like polymers, recently repeated by others, has thus far been limited to the use of canonical mononucleotides of RNA. Other groups have demonstrated polypeptide formation through wet-dry cycling.
Ooh,
More complex mixtures. I'm sorry, what?
I just want to make sure that my mouse quit working. What happened? Okay,
Yeah.
Go ahead.
More complex mixtures of polynucleotides and polypeptides present in protocells represent a next frontier in the testing of this hypothesis. In addition, there may be prebiotically plausible monomers that are close cousins to modern forms, some of which have been demonstrated to lead to polymers of use to biology, such as depsipeptides.
Yeah, depsipeptides. I like that
It's
one.
the first time I've ever seen that word. Well,
second time I've seen it, but I don't remember what it is. Okay.
And the use of alternative nucleobases. Also important is the consideration of the parameters that will limit the participating chemicals' availability due to decomposition or side reactions. A key next step is to observe the selection of the functional polymers described in section 3 by carrying out tests 3 to 7 below. Their presence and activity will be marked by an increased population of protocells in subsequent cycles. Analysis of protocell contents can confirm populations of these polymers and their byproducts.
Want me to take over for a minute?
Sure, go ahead.
Okay, 3. Encapsulated polymers must stabilize otherwise fragile membranous compartments having boundaries composed of lipid bilayers. We have proposed, Damer and Deamer, that first selective step initiating evolution in a population of protocells is that polymers interact with the membranes in such a way that the protocells become more resistant to environmental stresses, such as shear forces. As introduced in section 3, this S polymer is analogous to that of the cytoskeletal proteins that strengthen the membranous boundaries of cells today. In the absence of a stabilizing effect, fragile protocells will be continuously disrupted or simply leak their contents. Now that kind of folds into what I've been studying in intermediate filaments recently, and I'm looking at some of the possibilities of prototypical intermediate filaments by trying to get all of the genes and taking a look at how they could have formed. And if we had those intermediate filaments early on, I think the whole landscape changes if it becomes easy for a simple prototype intermediate filament. And I'm not reading the paper. I'm actually talking. I'm hypothesizing.
Ooh,
I'm using SpongeBob imagination.
that's
For-
right. Use your SpongeBob imagination.
Yeah, because that's actually a good thing. I don't know why they get so irritated by that. I like it. Another stress associated with wet-dry cycles is that ionic solutes become much more concentrated as Whoa.
Whoa,
I'm not going to do it.
whoa, whoa, whoa.
I'm not even going to try to wah that out. Wah, wah, wah.
Water.
Water. Oh, is that where he got it from?
That must have been.
Is lost. Yeah, it was this paper he was reading when he learned how to pronounce water. Water is lost to evaporation. When the dry membranes are rehydrated, a large osmotic gradient is established that causes the membranous compartments to swell as water diffuses across the membranes into the concentrated interior, potentially disrupting protocells by osmotic lysis. The osmotic pressure can be reduced if some of the encapsulated polymers can form ion-conducting defects or pores. P polymers. Interesting. Highlight that one. In the lipid bilayers. Oh, damn.
That allow.
In the lipid bilayers that allow the concentrated ions to escape. Pores have the additional effect of letting nutrients enter the protocells, which could support the emergence of primitive metabolic cycles, or M polymers. So, my mouse is kind of sluggish. I don't know why. Utilizing these nutrients, a significant observation by Vlasov et al. Is that RNA oligomers selected by in vitro evolution can produce ion-conducting defects in lipid bilayers. And let's see, Mayer et al. Have devised and tested a cycling protocell system operating at the phase transition between supercritical and subcritical gaseous CO2 in deep tectonic fault zones. I definitely want to highlight that, but I'm having trouble with my mouse. What could do that? They have observed that oligopeptides polymerize from amino acids and selectively integrate into the bilayer, there we go, bilayer structure of vesicle membranes. So that's what I was looking for this morning, thereby facilitating the mutual arrangement of their protection against hydrolysis while stabilizing the surrounding vesicle. Some of these peptides have also increased membrane permeability and protocell durability, effectively expressing the function of P polymers. A similar detection of the first P polymers emerging de novo in a wet-dry cycling system of protocells would be a significant milestone in the testing the hypothesis. I'm going to let you, I'm tripping on my tongue.
Okay.
Okay.
Sure. An important next prediction is that random mixtures of encapsulated polymers can evolve catalytic activity supporting primitive forms of metabolism. Bartel and Sostak demonstrated that 300 mers of RNA starting from entirely random sequences can evolve ligase activity by a process of selection and amplification. Similar experiences performed with encapsulated polymers will test the idea that catalytic functions can also emerge when protocells are subjected to selection through hydration, dehydration cycles. Number 6. At some point, sets of polymers must be reliably replicated to amplify functional polymers that emerge from initial mixtures of random sequences. The first informational iPolymer could be a simple templated replication system or resemble more sophisticated ribosomes as reported by Wagner et al. And Horning and Joyce. The complexities of cellular functions today suggest that chemical circuits operating in advanced protocells and progenote networks will also necessarily be complex and will be controlled by the well-tuned feedback systems mediated by F polymers. Emergent properties of protocells, number 7, by the way, may be observable in isolation, but are more likely to be accessible for study in an aggregate of many protocells, a progenote. Timeframes for bottom-up de novo in progenates, evolving interesting prebiotic properties could exceed the lifetimes of laboratory scientists and their institutions. It might therefore be advisable to jump the queue of random selection through the use of synthetic biology to
Okay, because that's one of the comments I was going to make is
create progenotes by introducing designed interacting polymer systems. Sorry, I had to finish the sentence.
Oh, okay. I'm sorry. Okay, so one of the comments I was going to make is that there could well be a million steps in this process, right? In the process at work. And we don't live long enough to find those million steps. So they're suggesting that we jump the queue a little bit.
Right.
Yeah.
Proof of concept. Yeah. To make shortcuts so that you can at least do various types of proof of concept.
And what are we going to be accused of if we jump the queue?
Well, it required intelligence to make happen.
There's no missing link. There's a missing link, right?
Yeah.
Okay, go ahead.
These programmed protocell populations, ooh, wow, what an alliteration, would then be subjected to cycles of selective pressures designed to test those polymer systems. Two recent examples informing this approach include RNA template copying inside protocells and vesicles containing an encapsulated enzyme that promotes synthesis of simple fatty acid derivatives stabilized to the magnesium positive ion.
Magnesium 2+ or whatever. Yeah.
Yeah. That makes me think it's an ion of magnesium required for ribosome activity and RNA synthesis. Continuing to cycle progenotes with the goal of testing whether evolutionary changes can be observed should use carefully crafted selective hurdles that reflect real conditions of hot spring pools and other prebiotically plausible environments. The emergence of depolymers controlling the duplication of sets of polymers and their delivery into daughter cells is probably inaccessible to experimental observation. Such a system may be too complex to be designed top-down by a synthetic biologist and may only be attainable via an accelerated bottom-up process of evolution. Perhaps this could be undertaken through the construction of an automated Genesis engine, sounds like a plot point in Star Trek, based on a miniaturized network of chemical robots similar to those developed by Cronin and co-workers.
Lee Cronin. There we go. Okay.
Given the challenges and costs involved, a second Genesis in the laboratory, long a theme of science fiction, will likely remain a scientific fiction.
Yep, and that is the problem, and that's why in 70 years, we still are where we are.
Yeah,
we got a long way to go, but I think with possible with computer simulations that we will be able to find the pathways. Because you see, the problem is that if we were to create these prebiotic conditions or some phase of this prebiotic condition, and we had enough different compounds, probably 100,000 different compounds turning around. Suppose that a self-replicating system did emerge. How would we know which pathway it took?
Right.
We don't have the ability to track every single molecule we have in the soup. And that is a very difficult problem. So either we work it out step by step, which takes forever. It takes about one paper for just, one step or a few steps, or we develop computer models that do it for us. And with computer models, we have the added benefit of being able to trap every state as we move.
Right.
So, with massive data storage, we can trap the states. And that's one of the things I'm looking at in Demeter's modeling is that, if I do these random models and I come up with a solution, how will I know which mix of random models unless I save all of that data at each stage?
Right.
That's one of the things we're looking at. And that's going to make it very costly in terms of computer cycles. And it's going to take a long time to run these things. So it's quite a problem. It's an engineering problem actually at this point. So
right.
Anyway, Should I read?
Go ahead. I can keep on reading if you'd rather.
Oh, I'd rather you kept on reading if you got
Okay. Section 7, actually, it's point 8, Section 7, 8.1, evolutionary implications of the hypothesis. 2 alternatives have been proposed as sites for the origin of life. Either life began in the ocean, perhaps using the energy and nutrients available in hydrothermal vents, or life began in hot springs associated with volcanic land masses that emerged from the global ocean over 4 billion years ago. The hot spring sites are analogous to those seen today in Iceland and Hawaii, supplied with fresh water distilled by evaporation from the salty sea and falling as precipitation. Given the 2 alternatives, salty seawater or fresh water on land, it is interesting to consider implications for life's early evolution and its long climb to complex forms such as land plants. The settings of these 2 scenarios are illustrated in Figure 9. Do you want to look at Figure 9 now or wait till?
Oh, let's see. I'm trying to figure out what page I'm on. Let's see. Let's see. I can go back to that. 8.3. Okay, I somehow moved ahead. Is this this is Figure 9 is what I use for the show icon, but we can look at it later.
Okay.
Okay.
8.2.
No.
We need to go to 8.2.
Let's see. Now I got to figure out where the hell I was.
I think we were just there.
We were
Now
just there.
we've already read that.
I don't know where I was. I got lost totally.
It's
Okay, there we go. Here we go. We're back. We're back. I'm on it.
Okay, if life began at submarine hydrothermal vents, its metabolism would at first have used the same energy as today's chemotropic and microbial populations. These organisms are anaerobic and do not require oxygen, respiration, or light photosynthesis. Instead, the energy source involves electron transport from a reduced substrate such as hydrogen or hydrogen sulfide to oxidants such as sulfate, ferric iron, or carbon dioxide as acceptors. The evolutionary path, therefore, requires that chemotropic vent life somehow made its way to the ocean surface where light was available, then discovered photosynthesis and evolved into the cyanobacteria that became a dominant form of life for the next 3 billion years. The switch to photosynthesis must have been necessarily rapid because as soon as the chemotropic organisms dispersed from the vent environment, they would be starved of source materials and energy in the much more dilute and cooler surrounding ocean.
Yep. And now, well, let's take a look at the illustration here, just for the hell of it. Okay, so here's the whole picture, sort of, from one end to the other. Freshwater eukaryotes, freshwater algae, land plants. Nice little diagram. I think it's the show icon. So, yep, that's what I used.
Cool. Yep.
Okay, so let's make it bigger again. And we'll try to get our shit together here at some point.
All right.
There.
Yep. Mm-hmm.
You know, there's too bad we don't have software tools. Okay, here we go.
I'm sure the first part was it is possible that life originating in shallow marine hydrothermal vents with some sunlight exposure might have a better chance to develop photosynthesis. A massive global culture of cyanobacteria in the ocean ultimately produced sufficient molecular oxygen so that the atmosphere became oxidizing about 2 billion years ago. At some point, eukaryotes emerged by symbiosis, still in salty water, and primitive eukaryotes in the shallow tide pools, began to form multicellular life resembling algae. Finally, this scenario suggests that around 500 million years ago, algae evolved the capability to survive when exposed to the atmosphere on mineral surfaces, and the first land plants emerged from seawater onto sterile continents. The alternative that life began in hot springs on land possesses a more direct evolutionary pathway to land plants. Freshwater life would also be chemotropic at first, depending on organic compounds available in hot spring environments through geochemical synthesis and extraterrestrial sources delivered during late accretion. However, early life was also continuously exposed to sunlight and could develop photosynthesis utilizing primitive pigments, such as PAHs as an energy source. Dry land exposure to damaging ultraviolet radiation would have been mitigated through hypolithic lifestyles.
Wow.
It might also have promoted key prebiotic reactions. Although salty seawater would have been an extreme environment for relatively fragile freshwater life, as microbial populations dispersed downhill towards the sea, they evolved cell waters with ion pumps to keep salt ions from disrupting intracellular metabolism. The volume of fresh water available was only a tiny fraction of that of ocean water, so when photosynthetic life was finally able to survive in the ocean, it filled a very large ecosystem niche. However, this marine adaptation does not mean that the evolution of freshwater microbial populations.
Let me get back up there.
Ceased. Okay. Instead, their evolutionary pathway paralleled that of marine life, but was driven by different environmental stresses. One was that hot spring water was continuously undergoing wet-dry cycles, causing 100-fold changes in the concentration of ionic solutes. So it was essential to develop cell walls to survive steep osmotic gradients. Another property of periodic wet-dry cycles is that at the origin of life, individual protocells were being brought into close contact during the drying phase of each cycle, forming symbiotic aggregates, progenotes. This enabled the later development of living multicellular consortia of microbial mats whose close collaborative sharing is a plausible pre-adaptation for the fusion events of endosymbiosis, which gave rise to eukaryotes. In the hot spring hypothesis scenario, subaerial landmasses on Earth were never sterile, but instead were continuously inhabited by freshwater versions of the microbial forms that later dominated the ocean. The proposal is supported. I'm sorry, what?
I just thought that was in the hot spring hypothesis scenario. Subaerial landmasses on Earth were never sterile, but instead, so basically life did not just leave the land, go into the sea, and then come back again.
Yeah, it was just there and then migrated into the ocean and it changed.
Okay.
This proposal is supported by evidence of stromatolites in the Pilbara in Western Australia thriving in subaerial hot springs 3.5 billion years ago. Paleosols carrying evidence of life 3.22 billion years ago have also been discovered in a preserved river floodplain in the Barberton region of South Africa. In addition,
I said, holy shit.
Yes.
Okay.
You're surprised by that statement?
Yeah. Okay, go ahead.
Yeah. Okay. Sometimes awesome things are said in these papers. In addition to an abundant supply of meteoric organics available, mute me.
Mute you?
Oh, too late. I coughed.
Okay, I'm sorry.
I wish I had a cough button, Well, I guess I did. I just didn't think to use it. Yeah, there it was. Okay. In addition to an abundant supply of meteoric organics available to the subaerial pools for life to start, continental rock weathering products such as phosphorus, crucial to early life and life today, are only available in an environment having a triple interface between water, atmosphere, and minerals.
Okay, I'll take over for you for a minute.
Sure.
Summarizing the arguments just presented, chemotrophic microbial communities originating in hot springs are continuously exposed to light energy during the day and therefore have an opportunity to develop photosynthetic metabolism. They are also transported downhill toward increasingly saline, and I went too far. conditions that force evolutionary adaptations to osmotic stress and turbulence at intertidal zones. These same land-based microbial communities continue to undergo a 3 billion year evolution to eukaryotic freshwater algae and plants, followed by fungi and protozoa developing in the shallow marine margins of continents. Now, some people say fungi and some people say fungi, and there is no consensus on this. So I like fungi.
We'll
Anyway,
have to write a paper on it someday.
Yeah. The earliest ancestor of land plants is currently understood to have been a form of freshwater terrestrial algae. In contrast, if the significant thermodynamic barriers for life to begin at marine hydrothermal vents were overcome, additional hurdles would still be encountered. The first would be the delivery from the vent environment to the ocean surface, where sufficient light energy was available to support the emergence of photosynthesis. The next is the journey into intertidal zones where eukaryotic marine algae and multicellular life would become possible by symbiotic mergers. Again, my intermediate filaments and some of their offshoots bind to different proteins and become the glue that holds cells together in mats and allow the beginning of multicellularity. So anyway, that's my comment.
Very cool.
The final hurdle would be adaptation to atmospheric exposure on mineral surfaces and the appearance of the first plants, such as primitive mosses that can use fresh water supplied by precipitation. Given that microbial communities have inhabited land as far back as the rock record can reveal, a direct evolutionary path from life originating in freshwater pools to the first terrestrial plants is perhaps a more plausible alternative than one in which life started at oceanic hydrothermal vents. And the vent people are going to hate them for saying that.
That's right.
Yep.
Them's fighting words.
We got a picture. Astrobiology implications of the hypothesis for the origin of life on other worlds. Continuously submerged environments such as hydrothermal vents clearly support highly adapted life forms of life on Earth today and might possibly harbor life on ocean worlds such as the icy moons of Europa and Enceladus. Plumes containing organic compounds and water emerging through cracks in I forgot how to say it again, Enceladus icy shell were discovered by the Cassini mission, suggesting that hydrothermal vents might be operating at a rock interface. I want to look at this picture. Oh, that's Enceladus. Okay. Deep. Rock interface deep. Oh, wrong mousey. Oh, my mouse is really driving me crazy. In an interior ocean. However, while potentially habitable due to their submarine setting and a lack of wet-dry cycling, these locations may not possess the chemical, thermodynamic, or combinatorial capacity for life to begin. Mars, on the contrary, has clear evidence of silica deposits, indicative of subaerial hydrothermal systems of a similar age as those discovered in the Pilbara. Surprisingly, this site at Columbia Hills, discovered by the Spirit rover, also resembles hot spring environments that preserve biosignatures of microbial communities on Earth. This discovery can guide future missions to promising places to search for signs of past life on Mars before it lost its surface habitability. I wonder if they had any input into what the current new...
I don't know. That would be fascinating to find out.
Right. Okay, I'm going to let you hit concluding remarks.
Concluding remarks. The hot spring hypothesis for an origin of life emerged from a fortuitous collaboration across multiple branches of science. Laboratory demonstrations supporting the RNA world hypothesis since the 1980s established an exciting direction in our field. However, despite the surprising catalytic properties of RNA molecules, we contend that the chemistry of life's beginning cannot be reduced to the action of a single molecular species, but instead required the interactions and selection of vast numbers of encapsulated polymer systems. These protocells were initially formed through processes of self-assembly of both the polymers and their compartments and evolved through the engine of a kinetic trap, subjecting them to combinatorial selection within cycling pools. This pre-Darwinian system of selection provided a scaffold for the booting up of the active molecular systems of life. Metaphors and insights from computer science have also informed the hypothesis by predicting that a collaborative network effect would occur in aggregates of protocells known as progenotes. Experimental work at multiple laboratories and more recently at field analog sites has accumulated a growing body of evidence. Finally, the discovery of 3.5 billion year old Archean fossil hot spring stromatolites of the Pilbara region of Western Australia provided key additional pieces to the puzzle. It possibly required more than 500 million years for the first fragile protocells emerging in the hydrothermal field pools to evolve into the
I'm sorry. Operator error. Okay.
To evolve into the robust microbial life that produced the durable fossil evidence of stromatolites. Somewhere along that timeline, the LUCAs appeared with a full complement of the functional polymers of life as we know it today. Note the purposeful use of plural in the previous sentence to indicate that LUCA was not a single cell from which all subsequent life descended, but instead would have been collectively sourced from widespread microbial mat communities that were sharing genetic information.
I'm so happy that you read that statement.
Yeah, it's a significant statement. I thought about what you had said before in our discussions as well.
Sal Cordova would probably choke on his chicken at this point if he saw that.
They don't exactly have a up-to-date view of evolutionary theory.
Yeah. He believes that the admission of LUCAs or multiple protein common ancestors basically is an admission that evolution can't happen, and it's actually just the opposite of that. It's just the opposite. Okay.
Forgel and Morowitz proposed that all living systems follow a principle of continuity in which new structures and systems emerge through variations on similar earlier forms. A final and perhaps the most remarkable implication of this hypothesis is that the earliest form life would have taken was an aggregate of collaborating units rather than competing individuals. Peering back through the lens of the principle of continuity, stromatolites are clearly the work of microbial mat communities. Perhaps those communities trace their ancestry to a simpler form of community, the progenote, which in turn got its start in deeper time as an aggregate of even simpler self-assembled protocells with encapsulated molecular systems. The tangled roots of the tree of life traced by intertwined protocell and progenote evolution may never be unraveled to reveal a clear picture of how life actually started on Earth. However, the hypothesis proposed here can be tested at several stages in the laboratory and in the field, and accumulating weight of evidence may then lead to understanding how life can start on a habitable planet such as the early Earth. The end.
Wow, fireworks. Anybody got a band? Because
I don't know what we should play.
What was that, 5 parts that we did? Was this a 5-part paper?
I think it's a 5-parter.
We did good.
Yeah,
so I just open up the stream in case anybody wants to come in. But anyway, the interesting thing about this is James Tour spent 13 videos talking about how this particular thing, this particular homogeneous set of molecules could not have happened because many other things could have happened, right?
That's right.
It could have been, basically racemic mixtures. It could have been, there's all these problems where you would end up with this sort of a soup or a tar, right?
Right.
Now, that is exactly the good news for abiogenesis. And so he spent 13 videos explaining how many other offshoot things could have happened, and we could have ended up with these complex mixtures inside of these progenotes, which would have been the perfect environment for a pathway to develop. Because if it had turned out that only one kind of compound could have been created, life would have been screwed basically at that point, because it would have went down one pathway, and all of the nutrients or all of the available molecules would have been just one thing, right? Homogeneous. And that is why they call me speed of pudding, because that is the exact hypothesis that I push against. It was not pudding. It was a very, very heterogeneous mixture of many, many different chemicals.
Right.
Anyway.
It was like Jell-O with grapes and little tangerine slices and coconut all mixed together.
Yep. And I want to point out here that these guys talk about sources, right?
Yeah.
Post this many pages of references for this paper. And I one section, I got 25 papers, I think, that I downloaded and I linked. So, I mean, that's quite a few papers. So,
and I'll bet none of them are linked to a blog.
No. There's what just happened? Somebody came in. I heard a beep. Then it went away.
I didn't hear a beep. I did like shift in my chair, but it didn't make a beeping sound.
I heard a real beep. Real beep. Yep. So let's go back and look at the top of this paper, and pat ourselves on the back. We published a very long paper, so I think we want to pick a shorter one next time. Maybe an offshoot from this, or
something foundational. Some important paper that kicked off a trend or settled a controversy or something like that. I don't know.
Yeah. Did they mention progenotes in the Bible?
Maybe in the Greek or something somewhere.
Hast thou sinned against thee?
Something like that, yeah. But here's that phrase again: seek first to understand, then to be understood. I like going through this because I had to read the content several times, and then now I can say I understand one hypothesis. Now I need to go and read the other hypotheses that are relevant to this. But have you wondered to yourself or come to a conclusion about when young Earth creationists are poo-pooing abiogenesis, what model of abiogenesis are they thinking about? Is it a particular one, or is it a distorted straw man version?
Yeah. I'm going through the illustrations here. Which one? It just doesn't. It doesn't carry a lot of weight. There's our gels.
Hello, Demeter. Are you
doing
well today?
Hey.
What did you think of our reading?
Oh, it was brilliant.
Oh, we are brilliant.
so-
Thank you.
Yeah, of course.
Don't go giving him the big head. He'll be insufferable.
Ooh.
Yeah.
I
So what I
already
can say, you ask what is the model of young Earth creationists for origin of life, right?
Yeah.
I can tell you right now what is their model. So their model is all of prebiotic blocks of life smashed each other against each other and formed fully developed organisms. This is their model.
Yeah.
So
Go ahead.
Yeah, so this is the model that they're constantly working with, especially with Angelo. I'm trying probably millions of times to tell him that this is not any of model that we are working, but he insists that we are working on that model that completely assembled at the same time all of the millions components and make a fully developed organism.
So isn't that kind of interesting how the reasoning makes our model be their model and require a God, basically. So I mean, they keep strawmanning our model so that we have to have a God because everything had to all pop up at once, and it had to just form. Nestling. By the way, I found Judea Pearl's book on causality, and guess what the dedication in the book was? It was to Danny, Danney.
So it is very amazing to see how many efforts are necessary to try to make some sense about origin of life. Because if you think about the science at all, I mean, nobody can at the moment. There is no single scientist effort that you can go and discover such a things as the origin of life, for example. Going back in, for example, beginning of 20th century, there was the first attempts of, for example, so-called a living molecular, which was considered that just one gene should be the living gene, something like that. But this is the beginning of 20th century. So nobody knew what actually gene is, but they knew that there is a gene. Around 1910 was discovered, the first viruses was discovered. And if you look at the Oparin's idea, Oparin, he proposed origin of life back in time around 1920, 23, 24. This is a very early, between the First and the Second World War. And still, what amazed me is that everybody, it looks like that everyone, a scientist, is correct in something and adding something new to the puzzle all the way up to now. And at the same time, everyone is wrong. So it's kind of interesting story that everyone is putting some extra puzzle to the. So Oparin, Miller experiment, 1953. After that, you have actually the discovery of DNA. Now we know what the information is. A few years later, the molecular biology, the central dogma of molecular biology appeared. So now we know how the information goes up and there. And so on and so on. And you see that it's a chain of many, many efforts, many events. And now you see, for example, this wonderful hypothesis by Deamer and Damer. It makes more detailed picture, makes more detailed. It never been such a detailed based on geology-wise hypothesis. So there are so many hypotheses which is just tube-like.
Yeah.
Tube-like my hypothesis is also tube style of hypothesis. But geology is important.
Yeah.
And if you look, for example, the paper which I wrote down, the point of no return paper.
Okay, you hear me now?
Oh yeah, Nestling.
Finally, I was trying to talk, but I couldn't get my mic working. All right. Like I wanted to say, like I like the analogy of like the blind man and the elephant. Like if you heard of it.
Yeah.
Like everyone comes from a different background and feels around the elephant, and they all come to different conclusions, which are correct in a way, but wrong in other ways.
Yeah, exactly. That's pretty much what it is.
Yeah. Even geology has just advanced, just greatly in the last 30, 40 years. And we're finding out, like watching some of these geology lectures by Nick Zentner, I think it is. Is that what his name is? Yeah,
Nick Zentner, yes.
Nicolana?
He does. He's talking about the geology of Washington and Oregon, and he basically, the thing that I took away from it is the massive amount of different environments that happen over time in geology. And some of them happen very, very quickly, with all these volcanic events and basically ice age floods and all of this catastrophic and then long weathering processes. I mean, there are so many environments that are presented that, and we're putting all of this together. And so all of this information is coming together, I think, in a lot of ways. But we're what are we, 50 years away? Yeah. I don't know.
Yeah, I mean, exactly. We need all of different fields, all of different science. Everything must be combined at last.
Yeah.
So we need chemistry. So all of the chemists that provide such a wonderful work, like a Sutherland,
Cronin.
and many other chemists, which provided lots of very clear hypotheses how the prebiotic blocks of life could appear. So we need those information. From another side, we need geological information, astronomical information, like this. We need molecular biologists as well to show some of interactions between RNA, peptides, and so on. We need also physicists. We need mathematicians. We need computer scientists. All of that is an effort. Not to mention, for example, two different approaches, which goes from up to the bottom and bottom from up. But both of those approaches is limited. You can, for example, if you go from up to bottom, you come to some domains, but that's it. You cannot go further to the actual real sequences.
Yeah.
And if you go from bottom to up, now you have another type of problem, which is from one side, if you're too loose in your hypothesis, you can bump your head in every single possible scenario, which might be many, and it will take forever. From another side, if you're too exact and too precise exactly what happened, you simply might miss many other scenarios. It's interesting. It's interesting.
Did you see the Norcell conference? Are you familiar with the Norcell conference? This thing that Bruce Damer told us about?
I didn't go there. I'm not very familiar with this.
Oh, it's a online conference, and it's happening. I think. When is it happening, Jerry? I forgot. I think it's happening very quickly, like at the end of the month, like the 29th or something like that. And it starts at 3 a.m., I guess, in our time.
Mm-hmm.
So I think it's 8 a.m. UTC or something like that. It ends up being 3 a.m. But we'll get you a link to that, and you can sign up for it. And I think they're also calling for speakers. So we need to get you that email and take a look at it. So.
To add to what Dimitar was saying, like I remember, and if you also remember Speed, like Eric Smith talked about ceilings and floors, where you have like on different levels, you have like a one system, like for example atoms, like the elements of the periodic table, giving rise to chemistry. And chemistry in turn gives rise to biochemistry and so on. And eventually you get also very complicated systems like genetics and other stuff. And in each level, you cannot, it's very difficult to predict them from the levels below. But still in each level, you need a science to describe each level of this hierarchy. So that's why and of course life intersects many different levels, and that's why you need many different fields of science to describe the whole process.
Is that Smith and Morowitz that you're referring to?
Yes.
Yeah. Okay.
I'm referring, like I remember, like when I asked you to play a clip from YouTube from Eric Smith. You remember that, the clip?
I don't.
Like it was with Danny. Like before he left, I asked you to play a clip and of a YouTube video, and then
Okay.
that was Eric Smith talking about like different levels of ceilings and floors within within nature.
Yeah. How long ago was that?
I think a month or something, maybe.
Oh my God, that's like, for me, that's like 2 centuries, or yeah. Do you have a link to that yet?
Oh, sure. I can look it up really easily.
Yeah, because it's probably.
Eric Smith is one of my favorite talkers about abiogenesis. You too, Demeter. You too.
Yeah, Demeter, I've told you about the Smith and Morowitz book. I don't know if you ever looked at it, but there's a lot of stuff in there. Have you seen that book?
Book I haven't read so far.
Smith and Morowitz, and the name of it is. Can you remember, Nestle? I have it right here somewhere.
It's a mouthful. I always can't remember the full name of it.
I have it in some stack of books somewhere, and I know not where.
It's The Origin and Nature of Life on Earth, the Emergence of the Fourth Geosphere.
Yeah, okay, that one. That is a good book. That is really worth checking out. But it is what is it? 600 pages of pretty tense reading.
It's really intense, really. Yeah, it's a bit intimidating for like newcomers to the whole. Like if you don't have a background or a basic background in biochemistry and genetics and such, it's really difficult to go into. Like you need to have a high school, maybe even a college level on understanding of this subject. So
yeah,
it might be difficult. It might be difficult for the layman. For the layman, I recommend Nick Lane book, The Vital Question, because that is much more reader friendly, so to speak, because he explains all things in more easily understandable terms.
Yeah. And then for the Hot Springs book, David Deamer has that book, Assembling Life, that he published last year.
Yep. Oh my God, Eric Hernandez is having a debate with somebody on YouTube. He's one of my favorite people to disagree with. He's the consciousness guy who thinks he knows what consciousness is. Basically, that it's got to be the soul, whatever. Okay, guys. What else can we cover?
I have the link here, like this. And also the time when he speaks about the different levels of floors and ceilings. Maybe you can play it again. It's just like a minute or so, not the whole video. Only a part. Only the end of it.
Also, Jerry putting access information about that Nordcell conference in there. You might want to capture it and put it in the description.
Let's see. Should I put it in the description? How public is that?
I don't know.
I remember my email from that you distribute with Bruce. So I know I can reach it. So okay. I can look at actually.
Yep. Let's see here.
From what I've read from like Nick Lane and Eric Smith, like they tend to favor the hydrothermal scenario that you just shared about.
Yeah.
So yeah, it's I also highly suspect that the alkaline hydrothermal vent is the cradle of life. Of course, that doesn't answer everything about the whole process. Only the beginning, from where it happened and what is the initial stress that it drove life into being.
The deep vent, the smokers.
More specifically, the white smokers. Yeah.
Yeah. So I agree with something, but I disagree with something. So as I mentioned before, it seems to me everything has something rational, something which maybe it's kind of more feasible. And at the same time, you have to pay attention. There are things that cannot happen at the same time, and that. So you cannot reject one or another, but you also have to be aware that something it's kind of going more natural attitude, let's say this way. Something, for example, alkali conditions are extremely unfriendly for RNA. You cannot have RNA in alkali conditions. That's it. Also, if you go as Bruce Damer said and David Deamer, you need some sort of kinetic trap to have a polymers. So something like dry cycle. So you have to go through the wet and dry cycle in order to polymerize.
If you want to finish first,
that could be compensated, of course. That could be compensated by super high pressure. But
not just that. Anyway, but if you want to finish first, then I can make my comments later. So.
Also could be compensated some sort of adsorption forces through the different rocks.
Right.
So you may have a different rocks which, from absorption point of view, they may create a super micro chambers where could be possible some sort of polymerization. So see, I'm not thinking in a way to reject something, but I'm thinking in a way what is more feasible, what is more likely to happen. So to me, hydrothermal vents are a very good source of organics. That's what I can say.
Yeah, about the wet and dry cycles, I of course, I don't really I'm really skeptical of that argument, because life itself doesn't use wet and dry cycles, obviously, to like synthesize polymers, right? So what happens is like water, like it is so basically the water paradox. Like in water, it's really unfavorable for polymerization. That's basically the argument, and you need like the wet and dry cycles to like force this reaction to happen. But in life, water is basically like really controlled within, like in a cell, it's really packed with proteins and other stuff that really reduces the activity of water, so to speak. Like water bound to a surface or of a molecule or something is behaves differently than free water in bulk water, basically. So and a similar thing can also happen in porous minerals, like the hydrothermal vent system, where you have like water, as you said, adsorbed to a surface, and that surface provides the environment where it can be still favorable for polymerization.
Yeah, but I agree. Yeah, it's true. But don't forget that the surface environment with hot spring, for example, are not mutually exclusive with the porous and adhesive forces. So you basically, for example, there is also in some of the scenarios for prebiotic synthesis of for nucleotides, for example, includes UV,
right?
UV forces. So I cannot tell you exactly details because I don't remember all of these formulas. But anyway, so there is a paper for that, and shows clearly that you need a specific moment where you need UV in order to convert and go through the nucleotides. But what I can say, the bottom line, and very important to take home lesson, you may have many scenarios, there is a many hypotheses, all kinds of ideas, but there is also almighty experiment. So almighty experiment will show likeliness of one way or another. Unfortunately, and I'm saying unfortunately, nobody funds too much origin of life research. There is just a few fund sources like NASA, for example, in Europe, ESA maybe, National Science Foundation maybe some funds a little bit of those kinds of research, but there is not much money there. unfortunately. So, and that is the reality.
Yeah, I think with the pharmaceutical industry is getting into this synthetic biology, and I think there will be more money in the future because of that.
True, but there is a shift of the goal of the aims.
Yeah.
So yeah, you can create a selective system, selective cycle, which actually is very popular. Back in time, Jacques Zostak demonstrated such a selective cycle, and actually by those cycles you can create ribozymes in laboratory conditions, where you can have ribozyme for almost everything that you want.
Yeah.
So, and well, make a ribozyme that cures something. Yeah, my point was we'll get there. Slowly but surely we'll be there. I'm sure there will be important experiments. From my perspective, two things are very important. First thing is the moment of information-dependent selection. Without this moment, there is no life. Without information-dependent selection or Darwinian evolution, nothing will happen further. Bruce Damon actually he proposed that. I mean, he also point exactly where will happen those kinds of events. So it's not like something that nobody knows. Everybody knows it. All of the serious scientists in the field knows very well. You must reach that point. Without that moment, there is nothing further. And that's one thing. And the second thing, I think maybe one of the next steps. I mean, there's many next steps. Okay, everybody thinks differently at the moment, but I think a very important moment, and it's feasible to be demonstrated a primitive translation. If you demonstrate primitive translation, how that happened, this is a huge step forward.
Yeah, that would be a big one.
Yeah.
Okay, can we play? In an hour and a half, we have Saturday Night at the Atheist, so I need a little break between them. So,
sure,
I can stop immediately. It's fine with me.
Okay, we'll play this and then head out. Is that okay?
Yep.
Sure.
Okay.
Good plan.
So let's see. So you had it bookmarked, right? That's it.
A bookmark. Oh, that's the moment where you can start. Yes.
Okay. Can you hear it? I can't hear it. Oh, I can't.
There's an important thing to say as part of building better scientific dialogue, which is the way that reductionism is used in biological sciences, which they would like to blame on physics, is completely different from the way reductionism is used in physics at its best. It may have been the way it was used in the physics of 100 years ago, but that's a thing that I wish we could get beyond, because the major accomplishment of the 20th century in physics was the understanding of the nature of emergent order. And we now realize that reductionism never made scientific sense without an understanding of emergence. Because if you didn't understand how a new kind of order can shield the details of smaller things, reductionism would lead you to need to know everything to predict anything. And the point of emergence is that you can have a predictive finite science at different levels, which lets through some of the details from the foundation and masks others. And the important thing is at the boundary where that happens, you have systematic ways of learning which details will come through and which will be masked. And a good reductionism in every science, I think, is going to have that dual character. It's the essence of reductionism is that if a composite thing is made of building blocks, then the number of combinations will be much larger than the number of kinds of building blocks. If all you want to know is the properties of the building blocks, then the number of experiments you have to do to get to answer that question should only be about as numerous as the building blocks. It should not have to be as numerous as all the combinations. And that's been incredibly useful in physics because it says we don't need to know everything about chemistry to work out the periodic table. And accomplishing an understanding of the periodic table is finite progress, even though we still don't know most of what's in chemistry. But you would never say that knowing the periodic table, we now know everything in chemistry. That would be stupid because it's manifestly false. So to understand the combinations, you need a new native language that is addressed to the characters of the combinations. And the knowledge of the building blocks does not give you that language because the combinations are a new kind of intellectual entity. So in biology, the role of reductionism is to know when you can solve part of a problem without having to solve the whole problem of the combinations. But in no mature science would you ever think that that means you don't have all the rest of that work to do. So we shouldn't think it in biology either.
Yeah, and that really ties in well with what we just read in the paper and what we were talking about as far as basically we're not going to be able to detail every step, but there are other ways to do it. And that is a brilliant piece on the tension between emergence and reduction. And I refer to something called constructivism or construction. That is the opposite of reduction, which I think also fits into all that. But anyway, that's a brilliant thing. I'll link that in the.
He's basically saying that like emergence and reductionism are often pitted against each other, but they are actually like complementary.
Yeah,
complementary
exactly.
to each other.
Right.
Now, I've also one thing. Of course, you it's no video, but I've one review paper that goes into the discussion about the water paradox, quote unquote paradox, like in the private chat. Maybe you can also share it in the comment section for people with who are interested, perhaps.
Yep.
So
let's see. Eric Smith. Let me put this video. Smith's video on reductionism. Okay, and I will put that in the description, and hopefully somebody will put it in chat. Maybe I'll put it in chat.
I can put it in chat also. But...
Okay. Hang on a second here. Let me get my stuff together, and then we'll end the broadcast so I can get a break because I'm old and feeble and, senile.
And I need to go to bed. See you later.
Yeah. Okay. We'll see you guys. Okay. We're going to end the broadcast, and I will link everything in the description that I can get a hold of here. And so goodbye, cruel world.
Okay, cruel world. Bye.
Goodbye.
Bye-bye.